Christoph Möhl from the German Center of Neurodegenerative Diseases in Bonn in Germany. And he's in the image and data analysis facility. And but he will present his work from his PhD and postdoc, PhD at Rudi Merkel's lab and postdoc at Heidelberg, EMBL, and
yeah. Thank you very much for the kind introduction, and also to the organizers to invite me here to this nice workshop. So my talk is entitled Quantitative Microscopy to Study Cell Migration. And I divided this in two parts. So the first part is called How do cells move? So studying single cell migration in cells in a culture dish. And this is more about the mechanics of cell migration, let's say. Yeah, and this I did at the Research Center Jülich in Mr. Merkel's lab. And the second part, Why do cells move, is about studying collective cell migration in living tissue, so during embryonic development at the Gilmer lab at EMBL Heidelberg. And this is more what is the function of cell migration, or the outcome of migrating cells in real living tissues. Okay, to start with the first part, how do cells move, or how to integrate heterogeneous live imaging data to one consistent picture. So here, this was my model system that I studied human skin cells, keratinocytes. You might have seen some other movies during the meeting of these keratinocytes, because they are quite popular to study cell migration and cell culture. This keratinocyte here, so both are imaged in bright field, but also in fluorescence microscopy. Here, a focal adhesion marker is expressed in this cell. So you see the focal adhesion, and here you see the actin cytoskeleton, and you see that during this migration process there is a lot of action going on in these both systems. Here, for example, for the focal adhesions, you see that the new focal adhesions are built here at the leading edge of the cell. Then they stay relatively stable in respect to the substrate, and finally they are disassembled here. So the cell crawls like over its adhesive site. Here this is a classical textbook view of how the cell migration machinery, let's say, works. It's described by Laufenburger and Torwitz in a very old review as a physically integrated process. And what they mean with this is that it's not like, for example, when you think about movement that is driven by flagella or cilia, that you can pin down the migration engine to one specific organelle, but it's more distributed over the whole cell, and then the interplay of different cellular structures drives the cell finally forward. So you have here the contractile actin-myosin system, and this is coupled to the underlying substrate with these adhesion sites, and the interplay of these two things somehow generates forces, and by this pattern of traction forces is built up, and this somehow drives the cell forward. So what we wanted to do is to spatially map these three main components of this migration machinery in a way that we can visualize how they are interconnected. So how is the interplay of these three components of the migration machine? The problem is that microscopic data is very heterogeneous, and it's not easy to average it over multiple cells or also over time. You see here 4 different keratinocytes imaged in phase contrast. and you see that they all look very different, and to even make it worse, they change their shape constantly over time. So, and yeah, the problem is now how can I integrate those very heterogeneous data? And so this is how we did it. So we first imaged a time-lapse series of adhesion dynamics. So the cells were transfected with a vinculin GFP. The vinculin is a marker of the focal adhesion, so it's a focal adhesion protein and coupled with GFP. This means it's fluorescent, and when you excite it with light, then you see the focal adhesion. And it was imaged with TIRF microscopy. It's a special kind of fluorescence microscopy called total internal reflection microscopy. And before I go on here with the cell shape normalization, how it works, I would like to show briefly why, so how this TIRF microscopy works, and yeah, why we used it. Here you see a scheme, a side view from the cell. This is the glass cover slip, the cell, and then we have here our vinculin GFP proteins, and these proteins are enriched in these focal adhesion sites, because yeah, this is a focal adhesion protein, but they are not only present there, but we have also a fraction that is diffusing in the cytoplasm. This is they are here constantly binding and unbinding, and you have some steady state where they are concentrated here in the adhesion sites, but you have them also in the cytoplasm. This means when you image them with normal epifluorescence microscopy, so you have to imagine the objective comes from below. Oh, sorry. So the objective comes from below, and what you would see here would be the focal adhesions, but also a very strong cytoplasmic background. Here in this TIRF image, you see also a background, but it's not so strong. So, and the trick with the TIRF microscopy is actually quite simple, that the excitation light to excite the fluorescence of these molecules comes in a very flat angle, and when the angle is very flat, then all the light is reflected here at the glass surface, and this means only how is it called? An evanescent wave is going here through the glass and only excites the molecules that are in very close proximity to the glass. In our case, that's exactly these ones that we want to see. So this is a very specialized method where you only image the fluorescence in very close proximity to the glass, and you don't need it that often, but if you want to study adhesion in a cell culture, then it's ideal. And you get a very good signal to noise ratio. Yeah.
Okay, because I mean, your previous movies didn't seem to have any adhesion at the back of the cell.
What do you mean?
You have very few focal adhesions at the back of the cell.
That's correct. Yeah.
And how does this go with this picture of kind of creating a...
Yeah, this shows us already that the picture is not really true. So it's a very simplified view, and I would say it's a standard textbook view here. This one. You mean this one?
Yes.
And I will show you in the end how it is really. So you have to look at in the.
I'm sorry.
No, I mean, but therefore it's a very good point.
Isn't it also that it depends on the cell type you're looking at? I mean, the keratinocytes do migrate completely different than fibroblasts, for example.
Yes.
Right, so
It's definitely
more a polarized shape with a very thin back, like contractile back. It's actively contracting.
I mean, it depends on many different aspects on the cell type, but also, of course, on the cells environment. So if the cells wouldn't have these flat glass structures, the migration would look totally different, of course.
So does turf limit your surface treatment options?
What you
Can
mean?
you put collagen or fibronectin on the surface, or is that too thick for turf?
No, that's okay. That's okay. But if you, for example,
Can't
do
use a gel.
if you if you add here a gel of 1 micrometer or so, it would be too much. Huh? I don't know. It's a few. I don't know exactly. Perhaps 100 nanometers or so.
Yeah, the evanescent waves 100 nanometers.
Okay. So, turf. Okay, cell shape normalization. So, yeah, I showed you that these cells have very heterogeneous shape, and we somehow have to bring our data together. What I did, I tracked over time the cell outline, and from the cell outline you can get the centroid of the cell, and then you get the track of migration, which is just the centroid then for each frame. And from that you can extract then the orientation of the cell. So the orientation is just where it moves to. So you get orientation vectors for each frame, and then we shifted the cell to a, let's say, cell-centric coordinate system, where this centroid is always in the origin from the from our coordinate system, and it always moves upwards. That means it's always moving along the y-axis of our coordinate system. And then our movie looks like this. Okay. Okay, and then we did a third step. Now the actual cell shape normalization step, where we morphed this cell shape to a circle. And then our cell looks like this, quite distorted.
Is that a linear elastic transformation?
This is a simple linear transformation. So you just take the radius here and then just linearly stretch or squeeze it. Okay.
Question. Why is it rotating so much? Because when I see these moves, I would expect that there would not be so much rotation in such an image.
Yeah.
Because the direction, when I look for a moving cell, it's quite stable.
Yeah, as I told you, so the orientation of the cell is dependent on this on the migration path, and the migration path is built with from the centroid of the cell, and when you have, for example, in this cell a small protrusion to this side, then it would shift a little bit in this direction and then go back, and therefore it's a bit rotating. You could also, of course, so there was some noise here in this migration path, and this are also smoothed. And if you smooth it more, then this back and forth rotation would be even less.
What if you were to monitor the nuclear position instead of the centroid? Would that give you a better or worse result? You can't do it with turf, but you could do a double imaging.
Yeah,
Off at the same time. Track the cell center, track the
yeah, of course, would be
but you expect it to be better than
would be more stable. Would be more stable. On the other hand, it wouldn't be that sensitive if a new protrusion would, for example, if a new protrusion would be established here, and then the cells finally migrate in this direction, it would, the orientation would come with a bit of delay, of course. But maybe, certainly it would be much more stable. Yeah.
could also do PIV and then try to establish a movement direction from PIV.
I think, I mean, with PIV you would have the problem that these don't move, yeah, and then the pattern is quite stable, and the movement is more growing because new adhesions are built here, and then others would be disassembled. So I think PIV would not work so well. Okay. When we have here our circular cell, so we can we have, let's say, we have normalized the cell shape over the whole time frame, but we have also also normalized our data over multiple cell, and then you can average this, and the most straightforward thing to average is just the image, just the pixel data.
Oh, excuse me, something. So you do the C image where you do the distortion. Do you also normalize for the size of the cell or just for the shape? I didn't understand that. So
Yeah,
the
so
F
the-
different sizes, right?
So the normalization works like this, that for each radius, so it's actually very simple. So you can, so you have here different distances to the cell outline from the cell center, and these distances are just linearly normalized to 1.
Is this actually equivalent, like finding the first spherical harmonic of the shape?
Right.
I mean, I can decompose the shape into spherical harmonics, right? And the first harmonic is the circle. I mean, basically, zero mode is going to be the circle, right?
Yeah.
So is this basically equivalent, or?
You could think of it like that, yeah?
So one important thing to have in mind here when you distort the image like this, of course, here the sizes, for example, of the adhesions are changed. They are distorted. Okay. So okay, now if you average this over the whole time frame and over multiple cells, then you get this image here, and this shows you in essence the density of the adhesion of the cell. And remember, this cell is always migrating upwards, and you see that the cell is clearly, or the adhesion intensity, the adhesion density of the cell is clearly polarized in respect to the cell migration. Because here at the rear you see a quite low adhesion density, as you mentioned earlier. You already saw it here in the one movie. And at the front you have a high adhesion density. What you also see here, that the adhesion is primarily present at the cell outline, so at the periphery of the cell. So you can do also other experiments. For example, you can stain your cells for actin and do exactly the same. And here you get the average actin density, and you also see that it's polarized. That you have here at the rear part, it looks more compressed, and here in the front part it's more spread out, and this part where I have the low actin density is where the nucleus is situated.
One question. So did you take those, I mean, did you average those images, did you take those images of actin with TURF or with confocal?
These actin images were taken with confocal microscopy, with standard confocal microscopy. So this means I not only see the actin at the adhesion sites, but in the whole cell. Afterwards, I show you actin movies where I image the actin only at the adhesion site, and we also bring it to this cell circle afterwards.
So is that a slice, or if you correct it for the thickness of the cell? Is that a density image or a total actin intensity?
I don't remember exactly. I think it's, I imagine it's a slice of, let's say, 2 microns or something like this. Okay, when you see these polarized patterns, what is now the interesting question is how these polarized patterns emerge, and therefore we looked at the dynamics of both the actin and also the focal adhesions. I first show you the focal adhesion dynamics. So I took exactly the same movies and detected the focal adhesion objects by quite simple object detection, so quite simple segmentation by intensity thresholding in essence. So when you have single focal adhesion objects over time, you can extract interesting features, like for example the area of the focal adhesion. And here in this plot you see just 4 example focal adhesions and how they change their area over the whole movie. So you see here, for example, this first adhesion starts to grow at approximately 5 minutes, then grows and then reaches a plateau and then shrinks finally. And this focal adhesion here becomes much larger, for example. And in one movie I have, I don't know, about 100 focal adhesions or something like this. These are just a few examples. And from the area profiles you can extract other interesting parameters, like for example the growth rate, which is just the change of area over time, or also the age of the adhesions. Another interesting parameter that I extracted was the sliding velocity of the focal adhesions. So it's actually not like this that they are always stable in respect to the substrate, but especially at the trailing edge they are sliding. Okay, then you can take this data and also transform to the normalized cell coordinate system and average. And here you see the results for some focal adhesion parameters. Here you have the focal adhesion area, and you see that that you have quite, with the beamer you don't see it so well, but here you have, at these lateral sides, you have the largest focal adhesions. The growth rate, you see very nicely that focal adhesions are only growing at the leading edge here. So the assembly rate is in red, and blue means negative growth rate, so blue means disassembly. And you see that after this assembly zone, you have directly a zone of disassembly. So many of these adhesions that are quite young are disassembling here. Here what you can also not so see in the projection is that at the lateral sides the growth rate is quite low, it's around 0. So these adhesions are quite stable. So this means we know these adhesions are stable and also large. And here at the rear you have then also a zone of focal adhesion disassembly. Here you have the sliding velocity, and you see that the focal adhesions in this disassembly zone here are sliding forward. They are somehow pulled inwards. Okay. Now I can compare this data with other experiments that are done totally independently. Of course, I took the same cells, but different markers. So in this case actin, so GFP actin, and here I imaged the actin in turf microscopy. I did this because we wanted to see how is the actin movement at the adhesion sites. And you see here, this is the detail view of the actin movement at the rear part of the cell, and you see this quite strong inward flow here, and we quantified this with an optical flow method that is optimized for actin flow, so for flow that is constant over time. And from this correlation-based method, you get then you obtain flow vectors for over the whole image. Yeah, and you see here the flow vectors for one cell. And we can again take this flow vector data, transform it, and average over multiple cells, and then you see here the generalized steady state actin flow pattern of our cell, and you see that the flow is very prominent at the rear of the cell. What is not so what is also the case that the flow is always inwards directed, which makes sense if you think that your actin-myosin network is a contractile system that always is pulled inward, let's say, and but here the flow, so in the zone of this adhesion assembly is disassembly is quite strong.
Question. The first thing in this picture, I would expect the actin flow to be highest at the front where the lamellipodium is. The fact that I do not see a high actin flow, does it mean that the cell is as fast as the actin flows?
No, no. The flow is relative to the substrate imaged, so not relative to the migration speed. Also, it's a different time scale. So this is this is micrometers per minute, I think, and the migration speed is much slower. And
one can't see from here, but the arrows at the front are pointing inwards or outwards?
you mean here in this picture? Here it looks quite random, but in the average image they're pointing inwards.
Inwards.
Yeah. Yeah, and I think the reason why we have this lower actin flow at the front is that we have at the front we have more adhesion, and therefore the force produced by the actin-myosin network is directly transduced through the adhesion sites to the substrate, and not goes in the flow, let's say.
So the actin flow has the same speed than the cell migrates?
No, it's much here it's much faster, and here I would say it's more or less zero. It's near zero at the front. I mean, this is also very cell type dependent. I don't know.
But you're also only seeing the actin very, very close to the surface.
Right.
There can be a forward
Right,
flow above that
yeah.
that you don't see with the surface.
Yeah, that's a very good point. We only see the actin very close to the substrate, and you can imagine when you think about this actin system as a gel that in the layers above you have more flow. That you have a gradient of flow which is very low at the
But even in your average picture, there is basically no actin at the front. Your average actin fluorescence.
You mean flow or actin density?
Actin density. There was, but it was low, yeah.
Yeah, it was lower because at the front you have normally this very flat lamellipodium, and therefore the actin density is lower there. Yeah. Yeah, here, this is now an overlay where I just overlaid the actin flow vectors with the adhesion density, and now you see this nice anti-correlation that I have high flow where I have low adhesion. So fast actin flow in regions of weak substrate coupling. Okay, then we also integrated traction force measurements. So the traction forces are measured like this, that the cells are seated on these elastic substrate, and if the cell exerts tractions to the substrate, it deforms the substrate like this, and in this substrate there are beads embedded, and then we can record the movement of the beads, and from the movement of the beads we can measure the deformation of the substrate. And if you know the elastic properties of the substrate, you can estimate the tractions. Okay. And here you see such a traction force experiment, the traction force vectors in red and focal adhesions in green. And you see now here, this is not image in turf, this is image in epifluorescence, because I have this thick elastomeric substrate beneath, and then I have a lot more cytoplasmic background. Okay. So we do this with multiple cells over a long time, and can average and get such a traction force pattern, where you see that we have the strongest traction here at the lateral sides. Yeah, and if you compare it now with our focal adhesion dynamics maps, you see that the strong traction forces are applied in regions where we have these very large and also very stable focal adhesions, which makes sense that these focal adhesions are able to effectively transduce the force to the substrate. Okay, another advantage of this method is that you can actively change the cell system, for example, by genetically manipulating the cells. Here we transfected the cells with a phosphorylation inhibited vinculin mutant. This means somehow the signaling pathway should be altered, and then you can compare your focal adhesion dynamics features, and where I saw then the largest differences was in the nucleation rate. So the nucleation rate is the emergence frequency of new focal adhesions. So when with this phosphorylation inhibited vinculin mutant, I have not so many focal adhesions we like in the wild type setting. Interestingly, other parameters like, for example, the sliding velocity are quite comparable. Okay, now to put all these pieces together, it looks a little bit more complex, like in this original 1D textbook view. We have here a zone where the focal adhesions are built. Then they grow a little bit, but a lot of them are disassembled here in front of the nucleus. Only a part of the adhesion grows further, and here these very large and stable focal adhesions are built, and then they are also finally disassembled. And in this region here of high adhesion, you have a relatively low actin flow, and therefore a strong inward flow at the rear where I have a low adhesion density. And here where I have my very stable focal adhesions, I have the largest traction forces, which somehow seem to squeeze the cell and push the big organelles like the nucleus forward. There were very similar results regarding tractions and actin flow described in these two papers. What they actually did, they did actin flow measurement and traction force microscopy at the same time at the same cells, which has some advantages because then you have really the direct link and not just comparing average images. What was essentially new in our work was that we added here the focal adhesion dynamics and this overall method of cell shape normalization, where you can, you can only display, let's say, the steady state of the cell or quasi-steady state. But the nice thing is that you can do a series of very simple and targeted experiments and then bring your data together, and you can do, and you are not limited here. So you can, so you could do now much more, like imaging microtubules, image keratin, image some proteins that are important for cell signaling, or maybe also measuring stiffness of the cell, and just do the same kind of analysis.
Wouldn't it, or I think it also would be possible to transform the cell not into a circle like we did, but also in this kind of shape, this ellipsoid shape, which is the normal shape of the cell.
Yeah. So this this is a good point. So maybe it would be better to first to first measure what is the average shape of the cell, and then to project it to this average shape. I mean, for these cells it's kind of okay, but if you think for neurons or something like this, you don't want to transform it to a circle, as at least not as a experimentalist. Okay. So with this I'm finished for the first part, and I would go to the second part. No, I'm not finished. So I want to thank the people who contributed to this work. So Norbert Kirchgessner, he had a lot of good ideas for the cell shape normalization, and also he did the traction force calculation. So actually it was his idea in doing this last step from the cell coordinate system and transforming it to the circle. Claudia Schäfer did the traction force microscopy and also the actin microscopy. Actually, it was like this, that I established these techniques with my focal adhesion movies, and then I just asked her if I can have her old movies, and also transformed all this to the system. Okay, and then I would like to thank my PhD supervisors, Bernd Hoffmann and Rudolf Merkel. Okay, second part, why do cells move? This is what I did then at the Gilmer lab at EMBL Heidelberg. There I studied collective cell migrations in living embryos. And during embryonic development, what happens, of course, that we have here an egg cell. So this is just one giant cell here. So this is the yolk, and this is the giant cell. And within 2 days, this one cell is transformed in a fully functional organism with, let's say, with everything. So with eyes, with brain, with muscle cells, bone, everything. And how this happens is that we have a coordinated interplay of cell divisions, of course, cell-cell adhesion and active cell migration, which in the end determines then the shape, the structure, and the organization of multicellular organism. If you only would have cell division, you would end up with a giant sphere. So you really need this cell adhesion modifications and active cell migration. So you see this also here in this time-lapse movie. So how the egg cell is dividing in multiple small cells, and then here this is the yolk sac in the middle, and here you have now the head of the embryo. Here you see the eye now, here this is the tail, here you see now the muscles develop. Now the muscles start to work, and finally after 2 days the embryo hatches. So, no. And this all in 48 hours. At the Gilmer lab, what we did is that we look at this embryonic development at a very late stage, so 28 hours after fertilization. And you see in this state the fish is almost there. So you see here the fish. And what we studied was this tissue here, what is called the lateral line. Primordium, it's a group of approximately 100 cells, and they move all the way from the head to the tail in around 10 hours. And you see this here in the detailed view, that while this cohort of cells is moving, it drops here these rosette-like precursor organs, and these organs will form afterwards the mechanosensory cells with the and with these cells the fish is able to sense pressure changes in the water. But the interesting thing for us was more that this is a very nice model system, how you see that you can see migrating cell cohorts, and then you have both, you have active movement of the tissue and also cell differentiation going on, and you can study how this is connected. Yeah.
Technical question. In the movie before, the fish was moving a lot. Am I assuming right that the fish was removed from the eggshell and was put in some agarose or something like that?
Yeah, I can show. I have a slide afterwards to explain. I think it comes now. Yeah. Yeah. Here with this slide, I just want to show the differences between cell culture imaging and whole embryo imaging from experimentalist view. And to sensitize you for the fact that this here is much more complex. In cell culture, it looks more or less like this. You have the cells directly attached to the substrate, and you look from beneath with your objective. Here in this case, you have the whole embryo, and as you said, it's the embryo is embedded in this agarose, and it's sedated, so it's sleeping and not moving. And the first challenge is now to express your GFP construct. Here, in the cell culture case, you are happy when almost all the cells are expressing the GFP. Then you have more samples. Here, in this case, it's different because you only want that a targeted tissue region is fluorescent. And if the whole fish would be fluorescent, the problem is that you don't see anything, or you see everything, and therefore you don't see anything. So this means that you have another challenge in genetic engineering. So you have to design GFP sensors that are specific. This means you have to know, you have to express the genes with a tissue-specific promoter region. And the problem is that this promoter region is hard to find, and normally it's done like this, that there's a blind screening approach, that you just express your protein of interest in a lot of regions of the genome, and then just see where it works or not, and then you know where your promoter region is more or less located. But then this DNA sequence is quite large, and you cannot do such easily genetic engineering, so loading gels and so it's much more complicated. Yeah, so just that you know that this differential expression is much more complicated than if you express it in just all the cells. Another problem is that, so here in the cell culture you have your cells directly on the glass. This means the working distance of your objective is very low, and you can just go for highest resolution objectives with all immersion that are available on the market, at least if you can if you can afford them. In this case you have you need somehow a compromise between a large working distance and a very good resolution. And this means here this is always higher resolution normally. This image here is largely over exaggerated, so so this would be a very, very bad mounting of the fish. Normally it lies really plainly on the glass, but then still you have a large distance, which is around, when you are good, then you have 18 microns to the primordium. Okay, so the resolution here is lower. But if you, for example, if you would like to image deep in the brain or so, it would be even worse. So, and there's also a reason why this lateral line primordium is such a beautiful model system, because it's still relatively close to the glass. Another problem is that your tissue is not perfectly parallelly aligned to the glass. So you have different working distances depending on where you image, and therefore in practice you normally image, for each frame you image a whole stack of images, and afterwards somehow project this stack. And this means when you do a time lapse you need very, very fast imaging. This means you cannot, normally you don't use standard confocal microscopy then, but so what I used was a spinning disk, but what would be also ideal, for example, a light sheet microscope where you can acquire your slices in a very fast time. Okay, so what I was essentially interested in was the differentiation of the cells and how this is related to the migration activity. When you look to the primordium from the side view, you have at the front the cells that are more flat, so they have more mesenchymal character, which means they are, let's say, more undifferentiated. And here at the back, where these rosette-like organs start to form, it's more an epithelial character, where the cells are higher, and also I expect that the cell-cell adhesion here should be higher. Here we have a side view of the primordium, so you now look from top of the fish. So you see two primordia on each side, and you see here that the flats here at the beginning are that the cells here at the front are quite flat, and then here they are higher. Okay, what I wanted to do in my experiments is to investigate this. So in this primordium, you have different levels of organization. Let's say you have first the tissue level, so the cell cohort, and as we've seen before, this tissue is undergoing a transition between epithelial and mesenchymal. Then you have the single cell level, where I measured the cell motility of single cells, so the cell shape dynamics, and then you have the molecular level, where I measured the actin dynamics, and I want to see how this is related. Okay, therefore I measured different parameters for the different levels of organization, and then in the end integrated. So what I measured here on the tissue level was just the thickness of the tissue. On the cell level, the lamellipodial dynamics, the speed of protrusion and retraction, and on the molecular level, the temporal change of the fluorescent actin signal. And I want to show you step by step how we did this. So for the tissue thickness, I just collected Z-stacks of these primordia in multiple fish, and they were labeled just with the standard marker, let's say with a membrane marker, but this membrane marker gave also a signal in the cytoplasmic area. And you see here the top view, the side view, and this view from the front. And here you see also that the tissue is not perfectly aligned. That always lies a little bit deformed in the in respect to the glass. And so what I had to do is to determine the axis of the tissue. So this is this would be essentially the height. And then just to register, and then what I did, because it was practically the easiest thing to do, to just register the image. It's maybe not the most elegant way, but quite computationally intense, but it was the easiest way to implement for me. And then after the registration, just I detected the tissue region just by intensity thresholding. And here you see the projections of my of the regions of the tissue. Yeah, and from this you can just, if you and from this you can just get the thickness of the tissue as a two-dimensional map. And you see here that at the front of the primordium the cells are rather flat, and then here they become larger. Okay, tissue thickness done. Next, lamellipodial dynamics and temporal change of fluorescent actin signal, and therefore I used the same raw data. So you see here now a model where, or a fish where all the nuclei of this lateral line primordium are labeled in red, and a random fraction of cells are labeled with live GFP, so with an actin marker. And you now see that this random labeling is quite useful here, because you can see here really the edges of the cell, and you can see the protrusions, and you can see also phylopodial-like structures. And this was actually very fascinating for me when I saw it the first time, because before I worked always with cell culture systems, and you are never sure if what you see is really true also in the real tissue. So when you see, let's say, the protrusions and retractions and the phyllopodia, but in fact it looks quite similar in the real tissue, at least here for this tissue. what I have to say is that this tissue is also migrating on a relatively flat substrate because it's migrating on a muscle sheet. And therefore it's perhaps also more comparable to cell culture system than in other tissues.
Why does it move on a line?
It moves on a line because you have here, so the muscle cells that are underneath are secreting a special growth factor. And it's not really clear how it works in detail. So if this growth factor directly influences the actin dynamics or the adhesion dynamics, or it's not fully clear, but it's clear that there is that you have here a sharp line of this FGF growth factor expressed or secreted in the.
But not by the cells in the.
Not by the cells, but by the underlying muscle cells, or not the muscle. Maybe there's another sheet of epithelial cell above the muscle cells. I'm not sure at the moment, but it's clearly for the underlying cells that produce this FGF. Okay, what I did from my experience, I actually did not image the whole sequence in the 2 channels, but I did one 2-channel image at the beginning and then just imaged the actin, because just it was very time limited. You know, I explained to you before that I always have to image a whole stack. And to have sufficient time resolution, I image a whole stack in 7 seconds and then image the next stack. So, and this is then on. Now the primordium is migrating in this direction. So, to confuse you a little bit, it's now migrating this direction.
Is there some selection of labeled cells on the surface, or are all the cells on the surface, or is that just an artifact of the way the reconstruction looks on a screen? And all of your cells look like the actin is touching the boundary with the outside tissue.
So
when you look at the nuclei, it looks like it's a 3D structure.
No, it's like this, that you have really one sheet of cells. So you don't have multiple layers here in this tissue. And once again, not all the cells of the tissue are labeled just randomly. Some cells. Yeah, okay, to extract the outlines of the cell, I did this with intensity thresholding. Yeah, I used here an approach. To be honest, I never used it afterwards, but here it worked quite well. You see here the intensity distribution of the whole image, and you have so the intensity of the pixels, and you see here 3 peaks. The first peak is the background. The second peak is the bleed-through of the red nuclei. I don't image them here, the red nuclei, but I also see them in my GFP channel because the filter settings are never perfect. And therefore, I have a really severe bleed-through, and this is actually the signal that I was interested in. So this is my actin. Yeah, and then I fitted here a 2-peak Gaussian function and just and then I set a threshold as a quantile of this function, and you see here how it changes over time. So I tried there a lot of different standard thresholding methods, but somehow in this particular setting this worked best. Yeah, so then I did some standard pre-processing, let's say, to close here these holes with morphological processing and remove some smaller objects, and then I finally ended up with these smoothed outlines here, and from these outlines so for the lamellipodial dynamics, from these outlines I measured then the protrusion velocities as vectors. Here, if you imagine this is the cell boundary at time point 1 and this is the cell boundary at time point t plus 1, then I quantified these displacement vectors, which is a little bit more complicated than it appears here. You cannot do such, you cannot do just a simple nearest neighbor approach. There are different methods to do this. Very elegant method from Till Brettschneider, which I actually didn't use because I was not aware of this when I did this. I did this here with the same method that I used to quantify the actin flow, just by correlation. So what I did, I produced images of the cell outline and then just made and then in principle with an optical flow method, with a PIV method, I got these errors, which in the end also works, although not as elegantly, let's say. Here you see now the results in green. So these are all my protrusion vectors here in green, and in red these are my retraction vectors. And protrusion means that during time the area is increased, the cell area is increased, and retraction means that the cell area is decreased during time. Okay. Lastly, we come to the molecular level. I measured the temperature change of the fluorescent actin signal. And as I told you, I used exactly the same raw data. What I first did, I fixed the tissue. So now it doesn't move forward anymore, but it's stuck. It's stuck in respect to the substrate. Then I just calculated the variation of the actin intensity over time. So just standard deviation over time and then normalized by the average intensity. And then you see here where you have strong changes in intensity over time. I only analyzed here the regions that are only the lamellipodial regions. So as I showed you before, I had the cell outlines, and then I just looked in 5 micron wide regions along the edge. And also time resolved, so I always calculated this temperature change over 7 time frames. And then it looks like this. And you see here these blobs appearing. This is basically the actin bursts that you see here. You have constant actin bursts coming up. And what I couldn't do, for example, was this actin flow analysis. This is just not possible in this embryonic setup. Okay. Now, to bring this data from the different levels of organization together to integrate them. I just map them, this time not on a 2D map, but just in a 1D coordinate system, just the distance from the leading edge of this tissue. And this is basically the reason why I needed this nucleus marker, that I knew where my cells were located relatively to the tip of the tissue. And here you see first the tissue thickness. So you see here you have the flat cells in the front, and then it increases, and you have here the thicker cells, epithelial-like cells at the rear. The protrusion velocity here in blue is higher at the front of the tissue, and then goes down, and is lower here in the more differentiated cells. Interestingly, the retraction velocity is stable over the whole tissue. And here you see this actin dynamics, so the temporal change of the actin intensity, and you see also that it's higher in these flat mesenchymal-like cells. Yeah, and then you can also do correlation plots between these parameters here, for example, the tissue thickness against the mean actin activity, and you see this anti-correlation that the flat cells have a higher activity than the thicker cells. Okay, to conclude, we observed a clear front-to-rear polarization in the cell cohort regarding the migration activity and the tissue shape. So this means high migration. So mesenchymal cells that are mainly flat show a high migration activity, while these thicker cells show a lower migration activity. Yeah, and with this I come to my end. I would like to thank the whole Gilmer group, especially Andrea Gruja, who is running the whole fish facility, Andreas Kunze, who teach me a lot of this genetic engineering stuff, which is quite challenging for the embryos. Céline Revenu made part of this life act construct that I used afterwards. And of course I want to thank the group leader Darren Gilmer. And I want to thank you for your attention. So are there any questions?
How do you keep the connectivity? How do you your cells are evolving in time, and you are mapping to a one-dimensional curve to follow. How do you keep the connectivity between two curves?
What do you mean?
If you go back to your slides.
This one?
No. Do one more. So,
you mean?
Yes, yes. You have, no, the other one, the next one. So you've mapped your boundaries. You've mapped your boundary to a one-dimensional boundary in relative to 2D.
Mm-hmm.
Yeah, and these are evolving in time.
Mm-hmm.
How do you do this, and how do you make sure that the connectivity in 2D is translated exactly into 1D?
You mean with connectivity that, for example, this point is afterwards?
Yes, yes,
there.
yes.
So with the it's actually not kept the connectivity. So I just measured these flow vectors. Okay.
But to get those measurements, you need to track the connectivity.
Yeah, okay, and this I did with this. Yeah, I think maybe I show you. I have some slides about this, then I can show it. So I did it with the same method as I measured the actin flow. This is for each point at the membrane, I have a template where I just look to this shape, let's say, okay, and then I shift this template around in the next time frame. This is the blue one. This is the next time frame, and make for each shifted template, I calculate the cross correlation of these two images, and what I get then is this kind of cross correlation map, and then you can just determine here the maximum, and then you have the shift. This is how I did it. And it's exactly the same method as I used for the actin flow. Yeah?
So what I liked is that in your system, the cells migrating in as tissue look very much like the cells migrating a single cell in cell culture. And there's a huge discussion going on whether the single cell analysis in cell culture actually represent the natural migrating behavior. Would you say that in your case that this is some kind of a special case because you're also migrating kind of in 2D, or would you say that many cell types migrating in tissue as a collective cell cohort might resemble many aspects you see as single cell in cell culture?
Yeah, I mean, I think this is a special case because they are migrating on this flat muscle sheet, and the muscles I think are also quite stiff. So, and I think it would look totally different if you see cells migrating through some mesenchymal tissue or some I don't know, some very watery collagen matrix, whatever. But still, I think there are many places in the organism where the migration looks exactly like this, because you have a lot of epithelia in the organism, and everywhere at these places, I think the cells migrate also like this.
I mean, maybe just a comment on that. If you imagine, I mean, like tumor cells in the tissue, they actually shape their environment. They reshape the collagen fibers so that they are like, that they can migrate on straight tracks actually. But I could imagine that there might be some relevance for that as well.
Mm-hmm. I mean, it's also like that these cells also, they also change their environment actively. So I told you that they are migrating on the stripe of FGF, of this growth factor, and it seems that they also degrade it. So they eat up the FGF, and maybe this also leads to this differentiation of the cells. That only the cells at the front are exposed to the FGF.
How does your framework handle cell division?
What you mean?
Can your cells migrating, can they divide?
Yes, they divide also, but but not that much. So let's say when the cells arrive, so when they start, you have around 100 cells, but when they arrive at the tail of the fish, you have only a few cells left. I don't know exactly, maybe 50 or so. But there's also cell division going on. So there are very nice movies of microtubules in these cells, and you then really see during the migration process how the spindles are formed, and then you have and then you have cell division.
I have a question to the first part of the talk.
Yeah.
So now you did the kind of spatial correlations between adhesions and actin. Did you do time correlations as well?
No time correlations at all, just spatial correlations. The problem with time is also that so as I said, the method is good for studying steady states, and then just compare. Timing is much more difficult. If you would like to see really timing events, so let's say you have an oscillation in the actin, and then you want to compare this with the oscillation of the traction force, you would need some synchronizing clock measurements.
I was actually more thinking about correlating the formation of adhesions at the front and the actin protrusions. Which in a sense, in one of your first movies, you kind of put in some actin, very little bit of actin protruding, and at the same time you had adhesion sites. You have this kind of in your movies.
There's also a nice paper from the Danusa group where they. Is this your work? No, okay. Where they do this time correlations, but therefore don't have the spatial correlation. But this is very nice. Where I think they did also independent experiments and then synchronized it somehow by always in each experiment calculating measuring the protrusion and retraction, and then taking this oscillation as a clock, let's say, and map all the other experiments on that. Okay.